This study came about because there has been a growing interest in the use of variant matched booster doses. In other words, third or fourth shots that were designed based upon the spike proteins of the newly emerging SARS-CoV-2 variants, which have begun to diverge pretty substantially from the ancestral strain matched vaccines. And it's been suggested that a variant matched booster dose may improve immunity and protection beyond that of a third homologous dose of the authorized vaccines. Now, we were particularly interested in Moderna's mRNA-1273 product, which had been developed in collaboration with the NIH's Vaccine Research Center, which is where I work under the mentorship of my primary PI, Dr. Daniel Duick, and the lead scientist on the NIH's preclinical COVID-19 vaccine program, Dr. Robert Cedar. And this is a good time to pause and highlight the fact that this study was tremendously large and came together very quickly. And this wouldn't have been possible without so many amazing colleagues and collaborators. Now, the reason for the immediacy of this work is that the recently discovered and now globally dominant Omicron variant has the greatest resistance to neutralization out of all variants so far. Now, in this study, we vaccinated rhesus macaques with a 2-dose prime boost regimen of mRNA-1273 at an identical dose to what is used in humans, 100 micrograms. Over the course of 9 months, total anti-spike antibody binding titers, and perhaps more importantly, neutralizing responses, decreased pretty substantially. We then boosted half of the primates with 50 micrograms of mRNA-1273 and the other half with 50 micrograms of mRNA-Omicron, which is matched to the Omicron spike. Both boosters increased binding titers and, most importantly, neutralizing titers, and in fact, increased neutralizing responses to Omicron beyond that seen after the initial prime boost regimen. However, we didn't notice a clear difference in the capacity of the Omicron matched vaccine to elicit anti-Omicron responses. Now, to drill down into this further, we then looked at memory B cells and their ability to bind the spike proteins of the ancestral strain, what we refer to in this paper as Washington 1, and the spike protein of Omicron. Now, the majority of memory B cells after the initial vaccine regimen recognized epitopes shared between both the ancestral strain and Omicron, although there were also some responses unique to the ancestral strain. Over the 9-month period, the entire memory B cell compartment contracted, and boosting with either vaccine resulted in expansion of the cross-reactive population. Although only the homologous mRNA-1273 vaccine was able to elicit a comparable expansion of memory B cells with unique specificities for the ancestral strain. Now, interestingly, we didn't see memory B cells which bound to unique sites in Omicron at any time point, even after the use of the Omicron matched vaccine. Now, to us, this suggests the possibility of a phenomenon known as original antigenic sin, what I prefer to call antigenic imprinting. In other words, the initial immune response to the vaccine was recalled by the use of the Omicron matched boost, although further analysis needs to be done to verify this finding. Regardless, both boosts were capable of expanding the critical cross-reactive population and increasing neutralizing responses, which was sufficient to provide complete protection in the lungs and thus protection against severe disease. I think the findings on original antigenic sin may stand out, but what's actually the most important here is that both boosts were able to provide complete protection against severe disease. And in fact, they increased neutralizing responses to Omicron and the beta variant, by the way, which I didn't mention earlier, beyond that seen after the initial vaccine regimen. And this suggests significant and continual antibody evolution, which is a finding consistent with other recent papers. And this is actually great news because this means that we don't actually have to wait for the optimization of new variant matched booster doses and further clinical trials. We already have the tools necessary to protect against Omicron and hopefully variants which have not yet emerged. Our conclusions are limited to our model system and study duration. Vaccination of non-human primates at these doses elicits stronger immune responses than what is seen in people. In addition, we only followed these animals for 1 month after the boost, which is when we challenged them. We needed to do pathology in order to determine the extent of protection provided by the vaccines. But unfortunately, that meant we didn't have these animals alive longer for further analysis or to do a second boost. First, it's necessary to further characterize the spike-specific memory B cell population. We need to sequence the B cells that bound only the ancestral strain spike, for instance, and determine exactly what the corresponding immunoglobulins can neutralize, and also determine if we were to switch the vaccine regimen to a variant matched boost such that that ancestral strain-specific population of memory B cells was lost, whether that would pose a problem if future variants were to emerge that are antigenically closer to the ancestral strain rather than to whichever variant we chose for the variant match boost. And second, our data would suggest that since the already authorized vaccines are sufficient to provide complete protection so far, that enhancing immunity, especially against asymptomatic infection or mild disease, may require switching the vaccine platform. There's a substantial interest in the use of mucosal vaccines, and I ultimately expect that to dominate the field in the near future.